Hi, everyone. Thank you for joining us today. Oh, yeah. Can everyone hear us? If you could reply in the chat real quick to make sure everything's good on our end. I saw something pop up.
Yes.
Okay. Hi, everyone. Thank you for joining us today for another APS seminar. It is my pleasure to introduce Maykon da Silva. Maykon grew up on a family farm in the south of Brazil. He did his undergraduate studies at the Federal University of Santa Maria, majoring in agronomic engineering. Currently, he is a second-year master's student in the Applied Plant Sciences program in the Agronomy Plant Genetics department. He is a member of the NAVE lab, working with management strategies for iron deficiency, chlorosis, and soybeans. He is the treasurer of the APS Grad Club and also on the organizing committee for the Plant Sciences Symposium. It is my pleasure to welcome Maykon.
Thanks, Lucas, for the introduction. As Lucas mentioned, I'm Maykon da Silva, a second-year master's student in the APS program, and today I'll be talking a little bit about my research project, which is titled Managing Iron Deficiency Chlorosis with Agronomics and Economics. So this is my presentation outline. We'll briefly go through an introduction and then move to the background, research objectives, experiment, preliminary results and conclusions, and then finalize with acknowledgements. To start, I would like to briefly introduce you to my project and state why it is an important study. So iron deficiency chlorosis is a soil-borne abiotic stress caused by a lack of soluble iron to the plant. IDC is prone on high pH soils and soils with large amounts of calcium carbonates, and its symptoms include interveinal chlorosis, stunting of the growth, and if the IDC is severe, necrosis. But why is IDC a problem? So let's imagine a soybean field affected by IDC in which symptoms are similar to these ones shown in this figure. How much do you think the soybean field is going to yield at the end of the growing season? There's your answer. IDC is a problem because most often its symptoms result in significant yield reductions. Only in the United States, IDC is estimated to cause yield losses totaling $260 million annually. So soybean IDC is complex and involves the whole plant. This means that being able to fully understand and efficiently manage IDC involves combining knowledge from several research areas, often segmented into breeding and genetics, agronomy, soil science, and plant physiology. Even though talking about all these areas together would be important to provide a complete insight into this nutrient stress, the focus of my master's project is on the agronomy part. So you'll notice throughout my presentation that this is our focus here. So there are several agronomic practices or management strategies that have been reported to prevent or reduce IDC losses in soybeans, such as tillage and drainage, foliar applications of iron, planting a tolerant variety, among others. But each of these management strategies come at some cost. Therefore, we designed our study to test 3 of the most often used management strategies for IDC in soybeans from a systems approach, which are variety selection, seeding rates, and iron chelates. But as I mentioned, each of these strategies come at some cost. So we know that there is a trade-off between managing IDC with varieties, with seeding rates, with iron chelates. So we don't want only to test these management strategies. We also want to evaluate these trade-offs in cost and yield in a way that we are able to inform farmers which of these management strategies will offer them the best return of investment. So background, soybean, origin, and importance. So soybean Glycinimax is a leguminous plant from the Fabaceae family, being of extremely value for livestock feed and human diets, primarily due to its protein-rich meal, as an oil seed crop for several industrial uses and food ingredients. Native to Eastern Asia, soybean domestication occurred about 5,000 years ago from its wild ancestor Glycine soja. In view of the several efforts in domestication, breeding improvements, and enhanced cropping systems, soybean has become the most cultivated and utilized oilseed crop worldwide. Currently, soybean is grown on more than 315 million acres worldwide, with a total annual production of more than 13 billion bushels. Brazil, United States, and Argentina are the leading countries in soybean production, and together these three countries contribute for 82% of the global production and 70% of the harvested area. Soybean was first cultivated in the United States as early as 1765, which cultivars were brought by sailors from China. Before the early 1950s, soybean was merely grown in the country as a soil nitrogen builder and as a forage crop. Then, two main factors triggered soybean production in an increasingly large scale in the country. These two factors were an explosion in livestock and poultry production due to low-cost soybean meal availability, and the recognition of its value as a source of vegetable oil. As we can see in this graph here, soybean production increased from about 300 million bushels in 1950 to more than 4 billion bushels in 2021. And this spike in soybean production occurred in the late 1950s. Along with the increase in production and harvested areas, a change in the geography of soybean production occurred as well. As breeding programs started developing varieties that were tolerant to drier conditions and that demanded less growing time, this expansion in soybean production has been associated with a northward and a westward shift in soybean acres. As we can see in this figure here, in these two figures, currently soybeans are grown in regions that it wasn't before, such as North Dakota, South Dakota, even more in northern Minnesota, western Nebraska, and western Kansas, and even Wisconsin and Michigan here. And how important was this shift in soybean acreage? Well, currently, more than 80% of the US soybean production is grown in the Midwestern United States. And Illinois, Iowa, and Minnesota are the top 3 soybean producing states in the country. But as soybean has been extensively grown in areas of the US Midwest, where high pH and large amounts of calcium carbonates are intrinsic properties of the soil, significant yield losses have been reported as a result of iron deficiency chlorosis. Using a geographic information system with soil pH maps, Hansen and collaborators, 2004, they mapped out the area where IDC is expected to be a problem in the north central United States. These authors, they estimated the total area of soybean grown under IDC-prone soils to be approximately 4.55 million acres in this region, encompassing the states of Minnesota, Iowa, South Dakota, and North Dakota. In addition, the authors estimated 160% expansion in soybean acreage in this region from 1970 to 2002, which is a fine demonstration of how IDC has become a major issue for soybean growers in the Midwest. But you might ask, where did these large amounts of calcium carbonates and high pH come from? Well, about 8,000 years ago, there was this huge ancient lake called Agassiz that is stretched from the corners of Minnesota, South Dakota, and North Dakota, and that covered a huge amount of land. In Minnesota and North Dakota, for example, the remnants of this lake, they form what's today is called the Red River Valley. Before tile drainage, these low depressional areas, they collected and held large amounts of water. With evaporation, lots of calcium carbonates and other salts, they remained, forming the called calcareous soils with large amounts of calcium carbonates. So in calcium carbonates in the soil, they act as lime, raising the soil pH. So that's the reason why in these regions we have soils with large amounts of calcium carbonates and consequently high soil pH. But what is IDC and what causes IDC? So iron is an essential micronutrient and one of the most required ones, as we can see in table 4.1 from Tyson-Siger. And iron is needed for several plant processes such as chlorophyll production, respiration, electron transfer, nitrogen fixation, as shown in these other images at the bottom of this slide. So iron is a cofactor for several plant processes. However, iron is the 4th most abundant mineral element in the Earth's crust. What does it mean? It means that IDC is not related to an absence of iron in the soil. The type of iron present in the soil solution is what determines availability for plant uptake and consequently the incidence of IDC. Soybean plants, they prefer to take up the reduced, the more soluble form of iron, called ferrous iron. However, when iron becomes less soluble or more oxidized in the soil, which means it is in its ferric form, soybean plants cannot absorb it. Without a sufficient supply of iron, chlorophyll production needed for photosynthesis and all those other plant processes that I just mentioned, they are restricted, giving rise to IDC, which is a common symptom of iron deficiency. So what is a definition of IDC? So iron deficiency chlorosis is a complex soil-borne abiotic stress and nutritional disorder caused by a lack of soluble iron to the plant. And what are the symptoms and timing of stress? In soybean, IDC is characterized by interveinal chlorosis, these yellow leaves with dark green veins, as shown in this image here, and stunting of the growth. Soybean plants, they remain short and they do not fully close the canopy. IDC symptoms, they appear as early as the first trifoliate emerges, as shown in this other figure here, and they can last until maturity if IDC is severe, but they are most common from V2 to V4. In addition, if IDC is severe, symptoms can include necrosis, as shown in this other image here, which can even cause plant death. But there are two major mechanisms that plants have developed to increase their ability to uptake iron from the soil when growing in iron-limiting conditions, which are strategy 1 and strategy 2. As a strategy 1 plant, soybeans, they rely on 3 major adaptive mechanisms to maintain adequate iron levels in its tissues. I won't talk too much about each of them, but first, soybean roots, they activate a proton pump called APS that releases hydrogen protons into the rhizosphere in an attempt to acidify this region immediately surrounding the roots, promoting the solubilization of ferric iron. Then soybean roots, they activate a ferric reductase enzyme located at the root cell wall that eventually reduces ferric iron to ferrous iron. Another mechanism, which is not shown in this image here, is related to the soybean's capacity to release chelating compounds or reducing agents, such as organic acids, that donate electrons to ferric iron, converting it to ferrous iron. Once iron is in its soluble form in the soil, it's transported into the cytosol through iron transporters. But what is important to remember here is that soybeans, they rely on the reduction of ferric iron to ferrous iron, because as I mentioned before, they prefer to uptake the soluble form of iron. Differently, strategy 2 plants such as corn, they release a phytosiderophore into the rhizosphere that binds with ferric iron, and this whole ferric iron phytosiderophore complex is then transported into the cytosol, which means that strategy 2 plants such as corn, they do not need iron to be in its soluble form in the soil. But there are several soil chemical factors and their interactions that change the solubility of iron in the soil and or that provide a difficult environment for these plant-mediated iron reduction mechanisms. And one of them is high soil pH. Alkaline soils with a pH ranging from 7.4 to 8.5, they precipitate iron as an iron hydroxide. The problem is that in the soil, iron hydroxides, they have very low solubility, reducing iron availability and causing deficiencies. As we can see in this image here on the right side of the slide of how soil pH affects availability of plant nutrients, we can see that with a pH ranging from 7.4 to 8.5, iron availability is significantly decreased. Another soil factor is high levels of calcium carbonates. Calcium carbonates in the soil, they act as a strong base, raising the soil pH and oxidizing ferrous iron to ferric iron, causing deficiencies. In addition, calcium carbonates, they also neutralize the excreted hydrogen protons and organic acids that were meant to convert ferric iron to ferrous iron, serving as a buffer against these plant-mediated iron reduction mechanisms. A third factor is residual soil nitrate. When soil nitrate is excessively present in the soil, soybean plants, they preferentially take up nitrate instead of doing biological fixation of nitrogen. And in this process, they release bicarbonate to balance intracellular charge. The problem, as I just mentioned, is that bicarbonate, they also serve as a buffer against these plant-mediated iron reduction mechanisms, neutralizing these excreted hydrogen protons and organic acids that were meant to convert ferric iron to ferrous iron. And a fourth factor is high soil moisture content. Wet soils, they are prone to IDC because in a limited air exchanging environment, CO2 released from respiration is trapped in soil water. The problem is that later this CO2 is dissolved, resulting in a buildup of bicarbonates in the soil. As I just mentioned, bicarbonate, they serve as a buffer against this plant-mediated iron reduction mechanism. And we can see in these two figures at the bottom of this slide here. In normal soil conditions, hydrogen protons or organic acids, they interact with ferric iron, converting it to ferrous iron, which is then absorbed by soybean roots. However, when we have iron deficient soil conditions, these hydrogen protons and organic acids, they interact with bicarbonate, avoiding the conversion of ferric iron to ferrous iron, which then cannot be absorbed by soybean roots, causing deficiencies. But let's take a closer look at Minnesota. In Minnesota, IDC is a major yield limiting factor in the regions of South Central, Southwest, West Central, and Northwest Minnesota. But why? As we can see in these two other figures here, the regions where we have high soil pH, represented by these pink and purple colors here, and the regions where we have large amounts of calcium carbonates at shallow depths, represented by this dark red color here, they overlap with the regions where IDC is mostly grown in the state. So again, IDC is a major issue for soybean growers in Minnesota. However, it's important to point out that while high soil pH and high levels of calcium carbonates are good indicators of high IDC risk, its prediction through soil testing is difficult and imprecise. Therefore, the best method to predict IDC is through past history. So let's see what soybean farmers have to say about it then. Hansen and collaborators, 2003, they surveyed soybean farmers in Western Minnesota, and these farmers indicated that IDC is a common and economically important issue in 99% of the farms that responded to their survey. These farmers also indicated that 24% of their crop is affected by IDC, and that on average, yield reductions of about 12 bushels per acre are associated with IDC. So again, IDC is a major issue for soybean growers in Minnesota. But how do these farmers manage IDC? In the same survey conducted by Hansen and collaborators, 2003, soybean farmers indicated that 90% of them make use of variety selection to manage IDC. 42% seeding management, including seeding rates and row spacing, 33% drainage, 16% tillage, and 11% fertility management, including foliar and soil applications of iron, and even iron as a seed treatment. And several of these management strategies have been reported to prevent or reduce IDC losses in soybeans by experimental studies. So let's get to the management strategies for IDC. So planting a tolerant variety has long been suggested as the most important and most practical strategy for IDC. Soybean varieties, they vary widely in how they respond to IDC, with some being highly tolerant and others being highly susceptible. But what is the difference between them? Highly tolerant varieties, they possess an increased capacity to reduce ferric iron to ferrous iron, increasing or enhancing iron uptake. Differently, highly susceptible varieties, they do not possess the same capacity to reduce ferric iron to ferrous iron. decreasing iron uptake and causing deficiencies. In a study comparing management strategies for IDC, Kaiser and collaborators 2014, they found that a susceptible variety produced the highest yield when IDC was not present. However, when IDC was severe, the tolerant variety out-yielded the susceptible variety by 34%. Therefore, the adoption of a tolerant variety is considered fundamental for profitable production in IDC-prone soils. But even the most tolerant varieties can experience yield reductions. Let's take a look at these two plots here from one of our field sites in Graceville, Minnesota. The only difference between them is that one was planted with a tolerant variety and the other was planted with a susceptible variety. And they were both planted with the worst conditions possible in our study. 0 iron chelate applying in the soil, nitrogen added to increase IDC intensity, and low seeding rate. And we can see that both of them were really affected by IDC. Then this question arises, well, what is the best way to manage IDC then? Overall, the best way to manage IDC is to pair a tolerant variety with another management strategy that minimizes losses. As farmers indicated in the survey, fertility management is another strategy adopted by them, mostly through the application of a chelated form of iron. But you might also ask, what is an iron chelate? So an iron chelate is a synthetic or naturally derived product that binds a metal cation, in this case iron, to multiple sites, as showing in these figures here, protecting it from interacting with other things in the soil and keeping iron available for a longer period of time in the soil. I highlighted EDDHA, DTPA, and EDTA because they are the most common forms of iron chelates utilized by soybean growers, and their effectiveness can vary substantially, but we won't get into too much detail about it. But the whole idea behind the utilization of an iron chelate is to keep iron soluble and available in the soil for plant uptake. The in-furrow applications of iron chelates have also been found to be effective in controlling IDC in soybeans. Kaiser and collaborators 2014, they found that the application of 3 pounds of EDDHA per acre significantly increased grain yield. Gamble and collaborators also 2014, they tested 3 rates of the same product, the same iron chelate, and they found that all of them were effective at reducing IDC symptoms under severe conditions. This is another photo from one of our field sites in Minnesota, in Graceville, Minnesota, and we can see how different plots look when we applied soybean and when we did not apply soybean. I just want to make it clear, soybean is a commercial iron chelate from CHS Inc. This is a very thick liquid that we apply in furrow in the soil. So in these plots in the left, we applied 4 pounds of soybean per acre, and the plots on the right side of the image, no soybean. And another way to manage IDC, as indicated by soybean growers, is through seeding management, mostly seeding rate, but it could also be row spacing and planting date. Ghost and Johnson, 2001, they tested 3 different seeding rates, half the recommended, and twice the recommended for the region where they conducted their study. And they verified that doubling the seeding rate significantly increased grain yield and reduced IDC symptoms of a susceptible variety. So another management strategy that is effective at controlling IDC. But you might also ask, what is the mechanism behind this population effect? So then we go back to the 3 adaptive mechanisms that soybeans have developed to increase their ability to uptake iron when growing in iron-limiting conditions. When we have a higher seeding rate, which means that we have a higher number of plants per unit of row, we have a larger amount of hydrogen protons and organic acids being released into the rhizosphere, which increases the reductive capacity and acidification of this region immediately surrounding the roots, which increases iron availability. There's more soluble iron in that part of the soil. However, when we have a low seeding rate, which means that we have a lower number of plants per unit of row, there is a lower amount of hydrogen protons and organic acids being released into the rhizosphere, decreasing iron availability. And there are also other management strategies for IDC that have been reported to prevent or reduce losses in soybeans that we won't get into too much detail because we don't have enough time, but they include tillage and drainage, growing a companion crop, and avoiding other forms of stress. So then we have our first research objective, which is to examine the trade-offs and interactive effects between varieties, populations, and iron chelate rates across a range of IDC levels. However, as I mentioned in the introduction, each of these management strategies come at some cost. For example, today's most tolerant IDC varieties, they come with some yield penalty relative to varieties grown elsewhere. Increased seeding rates, they significantly increase grain yield and reduce IDC symptoms, but soybean seed has become increasingly expensive. Iron chelates, they have been a game changer for farmers in the most heavily affected areas, but they can be quite costly at effective rates. So which one or which ones provide the best economic return? That's a good question. Not many studies have evaluated the relationship between their approaches or the management strategies that they evaluated and the cost associated with them. And the ones that did, most of them are quite outdated. So there is a need for this kind of evaluation. So then we have our second research objective, which is to develop an economic model informing soybean growers about return of investment for each of these management strategies individually and collectively to maximize economic returns across fields and farms. Typically, IDC is assessed in the field through a 1 to 5 visual rating system, popularly called green scores. As shown by this figure here, in such rating system, a score of 1 is given to a plot that is 100% green, where there is no yellow. And a score of 5 is given to a plot that is severely affected by IDC, where we have a lot of necrosis, a lot of chlorosis, a lot of stunting, and even plants dying or dead. Even though this is a simple method and it works quite well, there are two major problems associated with this visual rating system. It can be labor and time intensive, and it can be subjective due to the subjectivity of the human eye, which can introduce within and among raters variability. Then recently, image-based methods such as unmanned aerial vehicles, they have been proposed as an alternative approach to allow for greater efficiency, consistency, and precision. And they also work pretty well. However, most of these approaches, they are still categorical, ordinal classifications of IDC symptoms. And what intrigued us is that when we were evaluating or rating plots in the field, this phenomena occurred. We rated 2 plots as 2, as shown by these 2 figures here. They were both rated as 2. However, this one on the left here was almost a 1 because there was not a lot of symptoms. And this one on the right here could almost be a 3 because there was way more symptoms. This means that there is even though both plots were rated the same, there is a huge difference in terms of symptoms between them. And what intrigued us is how much this difference affects grain yield. That's another good question. We don't know. So then when going through the literature, we found that a more quantitative approach is needed to better understand IDC and to better understand how symptoms affect grain yield. We also found that IDC can affect soybeans even before it can be seen by the human eye. What does it mean? Well, when we go to the field and we rate IDC plots in a 1 to 5 visual rating system, maybe we are not considering those things that we cannot see. And maybe RGB images cannot capture things that RGB images don't capture. We also found that methods dealing with IDC should consider measuring chlorophyll content in a repeatable, efficient, and timely way. And that screening of cultivars for IDC tolerance should also include yield evaluation. So then we thought, well, what if we work on something that includes yield evaluation, and that is also a more quantitative approach to assess IDC? Well, while still looking through the research with the literature, we found that plant health can be quantitatively measured by calculating a series of vegetation indices. And that NDVI and NDRE vegetation indices, for example, they indirectly measure chlorophyll content. So then we thought, here it is. What if we try to work on a model to estimate grain yield based on timing and intensity of IDC with vegetation indices from drone imagery? But then the first thing we had to do was to verify if the variables we are trying to use to predict grain yield actually correlate very well with the variable we are trying to predict. So what we did, we extracted vegetation indexes values from our drone imagery or from our orthomosaics, and we ran some correlation analysis. And we found that NDVI from the drone, for example, correlates very well with grain yield. So that was a very good start for our third research objective, which is to develop a model to estimate grain yield based on timing and intensity of IDC using vegetation indices from drone imagery. Experiment, field sites, experimental design, and data collection. To accomplish the goals of our study, on-farm pair factorial experiments will be conducted at 3 locations in western Minnesota, where IDC, on soils where IDC has historically exhibited mild to severe iron deficiencies. These 3 locations are Foxham, Graysville, and Danvers. In addition, to vary the intensity of IDC, this experiment will be planted in 2 areas within each producer field, which we'll call site. So one site will be planted in a hotspot part in the field, which is an area where IDC is severe. And the other site will be planted in a neutral spot part in the field, which is a more representative part of the farm, where there could be some IDC, but where IDC is not as severe as in the hotspot. So therefore, in total, we'll have 6 unique field sites. And we'll test in total 24 treatments, representing the combination of all 4 main factors and their levels. So 3 levels of iron chelates, 0, 2, and 4 pounds of soybean per acre. As I mentioned before, soybean is an EDDH form of iron chelate, and this is a very thick liquid that we mix with water, and so we end up applying 0, 10, and 20 gallons of this mixture, soybean plus water, per acre. 2 varieties, a moderately tolerant and a highly tolerant variety, 2 seeding rates, 125 and 175,000 plants per acre, and 2 levels of IDC intensity by supplemental nitrogen. The application of supplemental nitrogen is an efficient protocol developed to increase IDC symptoms or IDC intensity and create a range of IDC symptoms. Therefore, in 50% of the plots, we'll apply about a pound of urea per plot, which represents about 69 pounds of nitrogen per acre. And in the other 50% of the plots will not apply nitrogen. Our experimental design will be a randomized complete blocks with split plot treatment design and 4 replications. In this figure here, I show how our experimental design was delineated. As you can see, the levels of iron chelate, they comprise each of the whole plots. Since we have 3 levels of iron chelates, we have 3 whole plots. And within each of these whole plots, we have 8 subplots, comprising the combination of each of the 2 varieties, the 2 seeding rates, and the 2 levels of IDC intensity by supplemental nitrogen. So as we can see here, this first whole plot here, we have 0 pounds of soybean, and for each subplot within this whole plot, we have the combination of the varieties, the seeding rates, and the IDC intensity by supplemental nitrogen. And here is, we have in total 4 reps, but I'm showing only 2 reps, just to show that here we have 2 randomization processes. The first one is within, for each of the whole plots. And then the other randomization process is for each of the subplots within each of the whole plots, okay? This experimental design, well, we'll have, the plot size will be 30 feet long by 10 feet wide, with 4 rows 2.5 feet apart from each other. This experimental design was selected to facilitate the logistics involving the application of iron chelate in furrow as a liquid suspension while sowing the trails. This protocol is the same protocol we utilized during the 2021 growing season, and which we plan to repeat this upcoming 2022 growing season. This is an aerial image from one of our field sites in Denver, Minnesota, and it's just to facilitate the visualization of how our experiment was delineated in the field. So again, these red rectangles here, they represent each of the whole plots. So since we have 3 rates of iron chelates, we have 3 whole plots per repetition. And the blue rectangles represent each of the subplots. So 8 subplots per whole plot, representing the combination of nitrogen, population, variety, and so in that specific level of soybean. All field sites will be planted with a precision research planter. Iron chelate rates will be applied in furrow as a liquid suspension with an in-furrow tubular system attached to the planter behind the seed tube that drizzles the product on the seeds. Nitrogen will be manually applied after planting when environmental conditions that minimize nitrogen losses prevail. Standard agronomic practices for soybean production in Western Minnesota will be used, such as for weed control. Each site will be truthfully characterized prior to and during the growing season regarding soil and weather data. Weather stations will be installed prior to planting in each of the locations and will provide average daily temperature and accumulated rainfall. Composite soil samples will be collected right after planting and will be sent for laboratory analysis for common soil chemical factors, micronutrient series, and nematode egg counts. Why nematode egg counts? Because Heterodera glycinis causes soil cyst nematode, and soil cyst nematode has symptoms very similar to IDC. SCN also causes yellowing of the leaves, stunting of the growth, and in severe cases causes necrosis. So we want to make sure that what we are seeing in the field is actually IDC and not SCN. That's why, that's because one of our varieties is actually susceptible to SCN as well. So we want to make sure what we are seeing is IDC and not SCN. After emergence, each plot will be weekly evaluated for IDC symptoms through a 1 to 5 visual ratings scale, popularly called Green scores, as well as ground-based NDVI and NDRE, and high-resolution drone imagery. These evaluations will occur from V1 to R6. The 1 to 5 visual ratings scale has been traditionally used to evaluate IDC symptoms in soybeans. In such rating scale, a score of 1 indicates that the whole plot is green. There is no yellow. A score of 2 indicates slight yellowing, most likely in the upper part of the canopy where IDC initiates. A score of 3 indicates chlorosis throughout the canopy, with most plants being yellow. A score of 4 indicates severe chlorosis, with all plants being yellow, and some stunting and some necrosis. And a score of 5 indicates most severe IDC symptoms, with a lot of necrosis, a lot of stunting, and even plants dying or dead. Drone imagery will be collected on the same days as visual scores with a MicaSense Red Edge MX sensor mounted on a DJI Inspire 2 drone. This camera captures 5 spectral bands almost at the same time. So blue, green, red, red edge, and near-infrared regions of the electromagnetic spectrum. Each flight will be autonomously performed with the assistance of Pix4D capture at a ground speed of 1 meter per second and at 23 meters above ground level. This altitude was selected to achieve a ground sample distance of 1.6 centimeters per pixel, which means high spatial resolution. All images will be taken from nadir view with 75% sight and front overlap. In addition, before and after each mission, images of the calibrated reflectance panel will be taken and will provide information regarding the light conditions during each flight. Also, a DLS, which stands for downlink light sensor, will also be mounted with the camera onto the drone and will provide solar irradiance. Why are these two things important? because we want to convert radiance to reflectance. Radiance is basically what the camera or what the drone sees, but that could be affected by atmospheric conditions. So what we want is reflectance. Reflectance is the intrinsic characteristics or properties of a surface or a target, and that's a derived value. So the calibration panel and the DLS, they are important to derive reflectance from radiance. Ground-based vegetation indices will also be collected on the same days as visual scores with the use of a crop canopy sensor called crop circle. This crop circle is an active sensor that provides NDVI and NDRE vegetation indices information, as well as basic reflectance such as red, near infrared, and red edge. This crop circle will be run approximately 1 meter above the crop canopy, only on the second row of each plot. And we will collect about 50 measurements per plot. Exact emergence dates and growth stages will also be recorded following the stage descriptions by Caveness and Thurr. At maturity stage are 8. The center 2 rows of each plot will be harvested with a small plot combine, and the weight of each sample will be converted to yield and adjusted to a 13% moisture basis. Upon completion of field activities, a complete economic analysis will be conducted in which we will relate grain prices and input costs into our analysis. So preliminary results. Let's take a quick look at the 2021 yield data. So I won't show data for all of our 3 locations because we don't have enough time, but I'll be showing data for Denver's and a little bit for Graceville, Minnesota, but I will explain why. So at Denver's, a 4-way interaction was found between site, soybean, variety, and population or seeding rate. Therefore, a separate analysis was performed within each site to test for treatment effects. I placed this blanked figure here just for us to go through each of the things that we'll be seeing here, just to facilitate interpretation. So on the y-axis, we have a response variable, grain yield. On the x-axis, we have both varieties, susceptible and tolerant. I refer susceptible to the moderately tolerant variety, but it's just to facilitate interpretation as well. On the right side of the figure, we have both seeding rates, 125 and 175,000 plants per acre. At the top of the figure, we have both sites, the hotspot site where IDC is severe and the neutral site where IDC, where there could be some IDC, but where IDC is not as severe as in the hotspot. And the legend represents the 3 rates of iron chelates. So let's take a look first at our neutral spot. So at our neutral spot, we didn't find any statistical differences in grain yield. As we can see, all treatments, they showed similar grain yields. In addition, IDC symptoms represented by the green boxes there, they didn't vary a lot as well, which makes sense. Not a lot of symptoms, not a lot of yield reductions, no differences. However, at our hotspot, we found 3 main things. First, without the application of soybean, increasing the seeding rate of the tolerant variety from 125 to 175,000 plants per acre significantly increased grain yield. So we went from an average of 32 bushels per acre to an average of 68 bushels per acre just by increasing the seeding rate. We can also see that IDC symptoms, they were very different as well. So we went from an average of 3 in our 1 to 5 visual rating system to an average of 1.62, which again makes sense. More symptoms, higher yield reductions. Less symptoms, higher yields. Second, we found that increasing the soybean rate from 0 to 4 pounds per acre of the susceptible variety at high seeding rates significantly increased grain yield as well. So we found a 22 bushel difference just by applying 4 pounds of soybean. So again, big differences. And third, we found that without soybean applied and at high seeding rates, the tolerant variety out-yielded the susceptible variety by 52%. So we went from an average of 44 bushels per acre to an average of 68 bushels per acre just by planting a tolerant variety. And IDC symptoms were very different as well. We went from an average of 3.12 in our 1 to 5 visual rating system to an average of 1.62. And now I want to show a bit of data from Graceville, but this is just to show you guys that different things were found for different locations. At Graceville, we also found a four-way interaction, but now between site, soybean, variety, and nitrogen. But why did I want to show data from Graceville? Because nitrogen in this case is very important. Why? So at our neutral spot in Graceville, we found that with the application of nitrogen, with a susceptible variety, and without applying soybean, we found a very significant yield reduction. Yield was significantly reduced. And why is this important? Soybean farmers, they won't go to the field and apply nitrogen in their fields. In Minnesota, most farmers, they rotate soybeans with corn. So last year was a very dry year. We have a very dry growing season. Farmers that applied nitrogen in their corn could have some remaining nitrate in the soil for this upcoming 2022 growing season. And why is this important? Well, even at neutral spots where IDC is not as severe as hot spots, farmers could have significant yield reductions if the correct management strategies are adopted. So if a farmer plants a susceptible variety at a neutral spot and there is nitrate carryover from the previous crop, without the application of soybean, he could expect significant yield reductions. So preliminary conclusions and further steps. So our preliminary results suggest different management strategies to be recommended depending the location and intensity of IDC, as I just showed. In neutral spots where lower intensities of IDC are found, treatments have less effect on IDC, or soybean yield, sorry. Differently, in hot spots where IDC is severe, treatments varied in their effect on IDC. So we saw that we can manage IDC with varieties, with seeding rates, and with iron chelates. But each of these management strategies come at some cost. Which one provides the best economic return? That's what we'll be working next. Relating grain prices and input costs into our analysis will enable us to provide farmers with recommendations based on what really matters at the end of the season: profitability, return of investment. Also, another goal of our project is to develop a model to estimate grain yield based on timing and intensity of IDC with vegetation indices from drone imagery. And that's something we are already working on, and that we should have more results sooner than later. And to conclude, I would like to acknowledge my advisor, Dr. Seth Nade. Thank you, Seth, for all your support so far. My thesis committee members for accepting being part of my thesis committee, Dr. Daniel Kaiser, Dr. Jeff Coulter, and Dr. Joe Knight. The Minnesota Soybean Research and Promotion Council for funding this project, the University of Minnesota and the Department of Agronomy and Plant Genetics for the opportunity of being part of this great graduate program, and the NAVE Lab crew. Thank you very much, guys. Victor, Jill, Mitch, Carlos, Junior, Anibal, Michael, and Regina. Thank you very much, guys. I wouldn't be able to do it without your help. And I'll take any questions. Thank you very much. Open the chat here. Oh, the chat is here. Oh, okay. I'll keep it here. Okay, I'll take any questions. Yes.
Variety of soybean varieties.
Which varieties? So Hussein asked which varieties we used. So the two varieties we planted in, which we'll plant this upcoming growing season as well. One is Asgrow Asgrow 12XFO and Asgrow 13XF1. Oh, sorry, Asgrow 13XFO and Asgrow 12XF1. Yes. Mm-hmm. And the 12XF1 is moderately tolerant to IDC, and the 13XFO is highly tolerant to IDC. Yes. Mm-hmm.
So IDC seems to be very spatially divergent. Will you account for ranges and rows in your experimental design to factor for the spatial variance?
So Lucas asked about the spatial variance of IDC and how we delineated our study to account for that. So that's one of the reasons we planted the same study or the same experiment in two areas within each producer field, because we wanted to account for those differences in each part of the field as well. But also we have 4 repetitions. We have 2 randomization processes. All treatments are 2 times randomized. So we should be able to account for that variability. And also we have 3 locations. So that should also help in regards to the spatial variability. Yes.
Maybe I missed it, but do you, how you decided to use that 2 different seed rates?
The seeding rates? Okay, so Kyunsu asked how we decided for the 2 seeding rates that we utilized. So in Minnesota, those 2 seeding rates, the recommended seeding rates for Minnesota, they are about 140, 150, 160,000 plants per acre. In 125 and 175, they are kind of the extremes that are still allowed for soybean growers in Minnesota for the insurance companies. So they are within the range that are allowed for soybean growers in Minnesota. So we basically selected the 2 extremes. Yeah. So low seeding rates and high seeding rates.
And those seeding rates, right, that trying to put this together too, it just had an impact on yield, not on the disease prevalence in the field.
As well, yes.
Oh, and disease prevalence in the field?
And IDC symptoms as well.
So like, what's that about?
Okay, so Amanda asked how the seeding rates, they affect IDC symptoms in the field. So as I mentioned in the during the background, soybean plants, they have developed 3 adaptive mechanisms to increase iron uptake when growing iron-limiting conditions. So they release organic acids and hydrogen proteins in the soil, which increases iron availability. So that means that they can uptake more iron, causing less symptoms. Yeah, so they will show less symptoms because they are absorbing more iron. Yeah, so I have questions here. So let me see. First, MJ, is there any reason why you use traded varieties over conventional varieties? Yes, so there are, well, the main reason that we use traded varieties is for weed control. We want to be efficient when we go to the field and we control for weeds. That's why we planted X10 varieties. And also because our farmers, like, they allow us to plant in their farms. So we also don't want to cause problems for the farmers. Sometimes they want to spray a herbicide, and we need to make sure that they won't kill our experimental studies. So that's why we usually select traded varieties. James Bowden asked another question. Do you think increasing the seeding rate is helping to release iron chelates, thus increasing yield? So iron chelates, they are not related to seeding rates. So higher seeding rates, there is a higher amount of organic acids and hydrogen proteins being released into the rhizosphere, but an iron chelate is actually a product that we applied in the soil. It's not something that occurs in the soil naturally. Okay. Nice one. Aaron Lawrence asked, why do you think the varietal effect was only apparent under the high seeding density treatment? Let me go back to the. Why do you think the varietal it's only apparent under the high? Okay, I think that the varietal effect was only appearing under the high seeding rate because as we can see here, at low seeding rate, the tolerant variety, the yield of the tolerant variety was also extremely reduced because not applying, just by planting, as I mentioned in the background, even tolerant varieties, they can experience yield reductions when that's the only management strategy we are adopting. And that's all we see here. When we didn't apply soybean and we had low seeding rates, we had significant yield reductions. So that's the reason. Even tolerant varieties can experience yield reductions when growing in a hotspot area, for example, without other management strategies along with it. I hope I answered the question. Maykon, could you provide more details on the prediction of yield using the vegetation indexes? Are the presented vegetation indexes values from a single type point or averaged across the season? So we flew the drone every week for almost 3 months. So we have vegetation indexes values for several time points during the growing season. And what we want to do is that we want to, basically the idea is to create a model. First, we want to create a model for each of these time points individually, and later see if we want to create a random forest or another machine learning algorithm that can integrate all these values from all these time points together to have only one model to estimate grain yield. So across fields, across field farms, for all locations, for all time points together. But first for each of them separately, and later on for all of them together, such as for example, a cross-validation process. Mm-hmm. Any other questions? Okay, so we'll be ending the meeting here, guys. Thank you very much for attending, and yep, thank you very much. Oh, okay. Okay. So
Nice
I should
job.
continue sharing, right? Oh, thank you.
Nice job. Yeah, that was an awesome talk.
Thank you. Yeah.
Maykon.
Nibo, thank you.
I have a sort of maybe not directly related question to you, but I just kind of wonder if you think that mixing, are there issues with mixing type 1 and type 2 plants in the same fields? Like you were talking about planting corn and it's dry, there's nitrogen applied and rotate in the soil. So I was just thinking about like you're doing like dual crops, right? Like could something actually exacerbate the problem?
I don't know about exacerbating the problem, but growing a companion crop, for example, helps alleviate IDC symptoms. For example, growing oats with corn, intercropping oats and corn, oats can uptake excessive nitrate from the soil, which could reduce IDC symptoms. Or even if it's a very wet year, they could also uptake this excess water, which could also help alleviate symptoms. But yes, let's say if you plant two crops in the same area and they both need soluble iron, that could exacerbate IDC symptoms in soybeans, for example. Yeah.
Can I ask a question about, if it's such a problem, why are they growing soybeans and not something else?
Well, because
I could ask.
yeah.
I drive out to Morris a lot.
Yeah, we asked that last.
Mhm.
Oh, did you?
So
Gary.
Western Minnesota and northern Minnesota is very dry. It doesn't rain as it rains in the southern regions of Minnesota. And even though farmers they grow corn there is not a lot more that you can grow there. So there are some farmers that rotate soybeans, corn, and beets, but there are not many options for those areas. And yeah.
So I wanted to rephrase that question that Emily asked, because I asked this last time.
Yes.
So why, I guess my question from Thursday was, why would soybeans be grown in Minnesota when they were introduced in the US if there was such problems with growing soybeans to begin with? I mean, I could see, okay, now we can figure out how to grow them, blah, blah, blah. And we can deal with some of the issues, we can deal with it. But why 70 or 80 years ago would soybeans have been a crop that farmers said, yeah, I'm going to grow this. And wow, it didn't grow very well, but I'm going to grow it again next year.
Yeah, well, I'm not really sure why they decided, but I'm pretty sure it's related to the amount of money you can make with soybeans. Soybeans, they can be very profitable. What else can you grow but soybeans? They will be as profitable as soybeans. I'm studying soybeans, so I have to,
Oh, that's funny.
Yeah, but that's what I think, they saw soybeans as an alternative, and that's why they started growing soybeans here.
Mm-hmm. All right, thanks.
Yeah.
So let's get back on this Vegetation thing. So I know North Dakota grows a lot of lentils and a lot of dry beans,
Mhm.
and they have a very different protein, profile than soybeans do, but
Yes.
do they have this, do all of the Fabaceae have this iron deficiency chlorosis issue?
Yeah, so other crops show it as well. Even sorghum that is not a dicot plant always also shows IDC symptoms. Yeah. Mhm. Yeah, but several other crops show IDC as well, symptoms as well.
Would how applicable would the models you're developing be to, like, say, a lentil farmer in North Dakota?
You mean with the drone imagery?
For measuring IDC, yeah.
I'm not really sure about other crops because we don't know, like maybe the way they reflect light is different the way they reflect, the way soybeans reflect light. So I'm not really sure if the model we are trying to develop could be used for another crop. Yeah, not soybeans. Yeah. Mm-hmm.
I think that's a very good answer. Lentils are a much lighter green color than wheat.
Yeah. Mm-hmm. Yeah.
How many tolerant varieties are there?
Not many actually. So this was, I can answer the way. So last week, they asked why the tolerant varieties, they come with some yield penalty relative to susceptible varieties. And this is something I was looking into. And one of the reasons is that breeding. So as I showed here, the total area of soybean grown under IDC prone soils, it's about 5 million acres, right? In the north central United States. But a lot more of soybeans is grown in regions that are not where you don't find IDC. So breeding programs, they really focus on other things and leave IDC tolerance kind of for a side project, and that's one of the reasons. Most of the varieties, they don't have high tolerance to IDC. Yeah, they are mostly susceptible. They focus on grain yield, they focus on other traits that could be more important for them in terms of profitability. Yeah.
Okay, can I ask a really ignorant soybean question?
Yes.
So when soybean farmers take their soybeans to the grain elevators, are they paid strictly on weight? Or is there a quality analysis that goes into like protein or oil or?
Yes. So one thing that I'm pretty sure they check is the amount of foreign material. That's the first thing. If your soybeans are super dirty, they will discount a percentage, is usually in percentage. I don't think that they are right now considering the amount of protein in oil in terms of how much they pay you. But I know that that's something they are debating. That could in the future, soybean growers that can produce soybeans with more protein or more oil, they could be paid better than other soybean farmers. Because, so Seth, my advisor, we do a lot of work in regards to protein in oil, and we have verified that in Minnesota, soybeans have less protein than soybeans grown in other states. For example, China, when they buy soybeans from the US, they kind of know that, oh, soybeans from Minnesota or this region in the Midwest, they have less protein. And soybeans from other states, they have more protein. So they are kind of debating those differences. Yeah, I won't be surprised if in the near future, they start paying based on the amount of protein in oil. But as far as I know, is not something that's occurring right now. Yeah.
I think that's a very interesting question. How does IDC affect soybean quality? You have all your seeds from your
Yes.
harvest. Can you send them to the NIR lab?
Very good question. That's something we did. We ran the NIR, we tested all of our samples, and we didn't verify any statistical differences in terms of quality. Protein, oil, even amino acids, everything the same.
Even
Yeah.
when they're highly impacted by chlorosis.
Even when they are highly impacted by chlorosis. Yeah, no
Isn't that
differences.
an interesting physiological study of trying to figure out how a plant
Yeah.
is in the process of, really.
Yeah, and I actually end up doing that because someone from North Dakota, they are studying that, and Seth asked me to run those analyses to actually see if there were differences, but we didn't find any. No, yeah. Mhm.
Well, you're really not in your final master's exam, so are there other, I feel like I'm peppering you with like.
No, I'm open to any questions. Yeah.
Comments about his presentation.
Really good. Very,
Thank you, guys.
you did like an exceptionally good job of like outlining what you were doing, why you were doing it every step of the way. Probably making that
Oh, thank you. Thank you very much. I was a little nervous, I have to say. Yes. Yeah. And I tend to speak a lot faster when I'm nervous. So yeah, I apologize for that. Okay, thank you.
I was listening to the podcast. It was awesome.
Oh, thank you.
Yeah.
Great. Very.
I like when you define every step of the way, kind of like simplifying the audience.
Thank you.
Do you know of the genes that people for IDC? Has that been characterized?
Yes, so several genes have been characterized. I remember when I took Aaron Lorenz's class a long time ago, I was still a master's student. I wrote a literature review about that. So as far as I know, there are maybe like five genes or 5 chromosomes that I think one is GM20 or GM5, something like that. Maybe Lucas knows it better than me, but they've been well characterized. I don't remember exactly, but yeah, maybe Lucas knows better. I don't know.
Yeah, there's MJ's whole project is focusing on one on chromosome 13. They're getting new student next year, Alina, will be focusing one on chromosome 5.
Okay, I thought your presentation was great. Can I be just a little picky?
Yes, for sure.
Metric versus English units.
Oh, mhm.
Like 70 pounds per acre is great if you're talking to farmers. Really should convert to kilos per hectare when you're talking to scientists.
Okay.
And so there are a few of them in there, and sometimes you said, like grams per whatever, and sometimes so just be consistent.
Yes, yes. And that's there's a reason for that. I had my units in international units or metrics. And then my advisor asked me to change it because he said, well, you're presenting for people in the United States. So that's why I changed. But yeah, it's.
If you were talking to a
right.
farmer group, you absolutely, they would have, I mean, many of them can figure out how to convert from
Okay.
kilos to heck.
Mhm. Yeah, but that's a very good point. Yeah. Next time I can, no, I'll keep the way.
Just know both.
Oh, both. Yeah, that's a good idea too. Yeah.
Just like anything we can do to get rid of like our stupid American units. Like just keep us going in the right direction.
Yeah.
Okay.
I agree.
Yeah.
Okay, here's another real, I mean, I'm getting picky now. Your
Yes,
presentation was
please.
really well done.
Yeah.
There was one spot on slide 42 where the specific epithet capitalized and it should have been lowercase. How picky is that?
40.
That's ridiculously picky. Oh, 40, no, it's 40.
Oh, sorry.
It's the nematode.
Here? Oh, here. Yes. Good point. Yeah.
It's sort
Right
of
here.
what I thought for
Yeah.
Amanda
Yep.
on her dry run.
Yeah.
Not the assumed word. Other words.
Oh, asshole.
I'm telling you, that's.
No, it's probably because I just copy and paste from somewhere and then I left that way. Yeah.
Okay, I told you I'm
Yeah,
getting-
thank you.
I
No,
had a
but
quick comment on that one as well. Is soybean cyst nematode, soil cyst nematode.
oh my God, yes. Soybean cyst.
Okay.
We all know what you mean.
Yeah.
I.
Oh my God.
Said soybean.
Wow. I will
So I
change
have,
it.
I have a question. I have a question.
Yeah.
This is
Yes.
not about your talk. So, and I think it was kind of referred to a minute ago, but I couldn't hear the question and I didn't really, so I couldn't, and then I think the answer was given by Lucas, but if you took those genes, those soybean cyst, or no, the iron deficiency chlorosis genes that Bob and Aaron are working on, would you be able, if you could put those genes into elite soybean lines, would you need to do any management sort of anymore? Could you do whatever,
So,
could you just plant those lines and be fine?
So as far as I know, I'm not a plant breeder, I'm not a geneticist, but as far as I know, there are no varieties or soybeans that are 100% resistant to IDC. So even though there are genes that confer or that provide some tolerance, none of them provide complete resistance.
Okay, all right,
Yes.
I got it.
Yep,
I have one more question for you.
yes.
So you're doing plant density. Do you, when you fly your drones, are you actually measuring plant density? I know you're planting differentially within a row.
Yes,
Are you then going through and like measuring plant density?
we are not doing it with the drone, but we did it manually. We went to the field and we counted the number of plants per, like 1 meter per row. We counted the number of plants to have an estimate. Yeah. And we verified those differences.
Yeah.
Yeah.
So the drought last year was pretty horrible.
Yes. So we had a
Just
few
behind this
problems. Yeah, in one of our field sites, some plants, they emerged later than others. I remember I went to the field and I opened the roll, many of the rolls, and we had plants that were already V2, and we had seeds that were still there intact. They hadn't emerged. Some emerged later on, but some never emerged. So that's why also counting the number of plants per, to have an estimate was important.
But can't you do that? I mean, I would think that you wouldn't have to do it manually if you're already flying drones.
Yes, we can do that with the drone. That's one of my goals as well, to compare.
A big goal of mine.
Yeah, to compare. But I've spent a lot of time trying to develop that model because we flew the drone many times. We have like more than 10 flights per field site. So in total, we have like about 60 missions. And they are big field sites. So lots of images, lots of things to process, lots of things to do. So yeah.
I was just trying to save you some time.
Yeah. Mhm.
I just want to echo what Emily said. I think that'd be really cool. Even if you showed a correlation to that, because you plant the population, but not all the seeds grow. I don't know what drone method you're using, but if you're using Austin's method, that's like step 1, classify plants versus soil, and then that plant tissue classified on scale of 0 to
Oh,
5.
the masking, the soil
So
masking.
if you do soil masking, you should be able to get that instantly, I think.
Yes. Mhm.
I was assuming that.
Yeah. No, that's not something that is difficult to do. I think I know how to do it. It's just like it's something else that I have to do. Yeah, that I haven't had enough time yet. Mhm. But that's a good point. It would be nice including it in another presentation. Mhm.
It would save some other poor graduate student coming after.
Oh, I was there too. I was doing it there. No, but yeah, that's a good point.
Any other comments for? Great job.
Thank you, guys.
Alright.
Yeah.
And Hussein's giving his dry run on Thursday. Remind Gary he's coming. 'cause I'm going to be-
I will be there.
Okay, he said he will be here. Yeah.
So my group has to be here on Thursday with Gary instead of me. All right,
So we will
Sounds
not-
good.
that's great. You will. Amanda won't, she'll be at a meeting. See, I do pay.
Thank you, Gary.
Oh, thanks,
Can I
thanks a
end up
lot.
the meeting here?
Yep, you can go ahead, end it.
Okay, thank you. Bye-bye.
All right, see ya.
Oh, well, we could stay talking about this.
I thought that was so good.
Welcome, Ollie. What are you doing? Oh, I think there's in high school and college now. Must do a lot more presentations than we do. They're all really good. Even the weak students are like better than the best students that I remember.